Hello everyone, good afternoon. Thank you for watching my previous video. In the previous video, we have learned about the cell membrane and its receptors. Now we are going to learn about membrane transport. Transport across membrane. Membrane transport based on ATP requirement. That is, energy is required or energy is not required. If energy is not required, that is known as passive transport. If energy is required, what do we call it? It is an active transport because we are using ATP, which requires ATP is active. Energy is required is active. Energy is not required. No energy is required. Passive. Okay. And under passive transport, we have three main transport mechanisms, which are the what are those? Simple diffusion, facilitated diffusion, osmosis. Simple diffusion, it is based on Fick's law and lipid solubility. What is Fick's law of simple diffusion? More the concentration gradient of the molecule, more is the diffusion. Like on the outer side, if sodium is more, it tends to diffuse inside because outside it is more. Potassium, more on the inside, it tends to diffuse outside. Okay. And diffusion is inversely proportional to thickness of the membrane. That is, more thicker the membrane, less is the diffusion. It's same like more thicker, less thicker the membrane, less is the diffusion. Both these comes under the Fick's law. And as you know, the cell membrane is made up of lipids. So more is the lipid solubility of a particular component, more is its solubility, more easily it diffuses outside. One more thing, diffusion is inversely proportional to size of the molecule. More bigger the molecule, less is the diffusion. Less is the size of the molecule, it will easily diffuse through the crevices. Okay, facilitated diffusion. Under passive transport, we have another diffusion, facilitated diffusion. You're using some facility present on the cell membrane, which are known, what are those? Those are the transporters. So that is known as facilitated diffusion. But that facility does not using that facility doesn't require any ATP, so it is passive facilitated diffusion. For example, in the question, if it is given as uh, only in options, it's only facilitated diffusion, you have to mark facilitated diffusion. If facilitated diffusion is not there, you have to mark it as passive transport. Okay. Examples are glutes. Your glucose transporters, GLUT1, GLUT2, GLUT3, GLUT4, GLUT5. Where is GLUT1 present? RBCs, brain. GLUT4 is important. GLUT4 is dependent on insulin. It mediates insulin mediated glucose uptake. Uptake of glucose is based on GLUTs. Aquaporins, those are transport of water molecules across the membrane. Okay, and one more thing you have to remember this graph. Since in facilitated diffusion there is a carrier will get saturated at a point. So the graph will come out like this, parabola or something. And the straight thing is the simple diffusion ka graph. Okay. One more passive transport mechanism is osmosis. Normal plasma osmolality is 280 to 300 milliosmoles. Sodium and water is required for osmosis. Water always follows sodium. And when we use ATP for the transport, it is active transport. Active transport can be primary active or secondary active. Okay. Secondary active can be further divided into symporters or co-transporters and exchangers or anti-transporters. Symporter or co-transporters, it will take the molecule, both the molecules either inside or outside. Exchangers is one inside and one molecule outside. I have explained active transport in the next coming slide. Okay. Active transport. Primary active. Primary active has three main examples: sodium, potassium ATPase, calcium ATPase, H plus ATPase. Sodium, potassium ATPase is important. Even the structure of it is important. The sodium and potassium ATPase consists of two subunits, alpha subunit and beta subunit. It is known as the since it has two subunits, it is heterodimer. One alpha, one beta. Therefore, heterodimer. Dimer because two subunits. If there is two alpha, it will become homodimer. But sodium potassium ATPase is a heterodimer, consists of one alpha and one beta subunit. It transports sodium outside the cell and potassium into the cell. Remember, sodium always floats in a salt solution, sodium chloride solution. So major electrolytes on the outer surface of the cell is sodium and chloride. On the inner side is potassium, bicarbonate, magnesium, everything comes inside. Major electrolyte outside is sodium, major inside is potassium. Okay. Alpha subunit has the binding site for sodium and potassium. This constitutes the this this sodium potassium ATPase contributes to the basal metabolic rate. And one more thing, the coupling ratio is important, which is 3 is to 2. That is, for every 3 sodium moving out, 2 potassium come in. This is known as coupling ratio, which is 3 is to 2. Okay. Secondary active transport, it is usually two or more substances are transported. These are co-transporters and exchangers. Co-transporters, example is sodium glucose co-transporter, also known as SGLT. Sodium potassium 2 chloride co-transporter, also known as Baptin channel. Sodium iodide importer. So where do you see these three is also important. Sodium glucose co-transporter, known as SGLT, is seen both in intestine and kidney. SGLT1, write it as I, present in intestine. 2, since we have two kidneys, it is SGLT2 is present in kidney. Remember like that. And within the kidney, particularly, it is seen in PCT. You can also remember SGLT as instead of sodium glucose, secondary glucose transporter. Because you have a question, where do you find secondary active transport? SGLT. And where do you find, which channel do you find, which secondary active transport channel you find in PCT? That can, then answer will become SGLT2. Okay. Sodium potassium chloride 2 chloride co-transporter. This is also known as Barton channel. This channel is present in loop of Henle. The mutation of this channel leads to Barton's syndrome. This channel can be inhibited by loop diuretics. Okay. Sodium iodide importer. Where do you find iodine? Iodine is important for thyroid gland for production of thyroid hormones. So sodium iodide importer is seen in thyroid gland. Okay. Under secondary active transport, we also have exchangers. Chloride bicarbonate exchanger, which is seen in RBC. Okay. Which is required for this release of oxygen. Okay. This is all for today. Remember this flow chart and remember these. These will help you to solve at least 2 to 3 questions. In this, basic definitions of passive and active transport is important. Fick's law, lipid solubility. This graph itself is a question. Under secondary active transports, importers, co-transporters, exchanges, anti-transports, all these examples. This slide is most important, and this one is also very, very important. Okay, that's all for today. Thank you so much. Keep supporting, keep sharing.